We're beginning to understand the causes of the main developmental brain disorders, such as, for example, epilepsy or psychosis even. We now know that the environment, but very particularly changes in specific genes, can actually be related to the onset of these conditions. However, genes per se, do not cause this disorder. Genes contain the information that is required to build the brain. And therefore, we need to understand how these variation in genes or in the environment actually impact human brain development to be able to, understand the causes and treat these conditions. Traditionally, we have used mostly animal models to these experiments. However, there are aspects of the biology of humans that are unique and that cannot be reproduced in animal models. So at Keynes, what we're trying to do is to bring together all the experts we have on human brain development to tackle this problem. We have, world-leading experts in human brain imaging that can, look at great resolution on living embryos and fetuses and children and how the brain develops using imaging techniques. But we are now becoming smarter a bit about using cellular models to be able to, in the lab, being able to reproduce some of these aspects. For example, we can use tissue from embryos to gather some properties of the developing brain. We can even use brain organoids, which are, somehow, small avatars or small models of human brain development that we can actually manipulate to understand how these brain conditions are arising.
My research aims to understand how the human brain and its activity first develop and become organised at the time of birth. This research is motivated by my other work as a consultant in paediatric neurodisability in the Evelina Children's Hospital, where I manage the care of babies and children who unfortunately have difficulties because of an early brain injury. We use techniques like magnetic resonance imaging or MRI scanning, which can safely provide us with very detailed pictures of the brain, which we can then study with methods like computational analysis or using mathematical models. We're trying to understand when particular structures and patterns of activity develop in the early human brain and how this development is influenced by different factors like injuries or preterm birth. And we found that even in the womb or as a newborn baby, the human brain is already highly organised in terms of its connections and activity, and that when this is altered, it can predict then how a child might do later in life with their development. Ultimately, we hope the research can really help us to understand why some children have difficulties, and that will mean that we'll be able to give families more accurate information about diagnosis and how their children will develop in the future. And this, we hope, will help us to guide us then how to treat them more effectively.
My group studies how the human brain develops to the right size and shape. And we're particularly interested in how you go from this very early, smooth structure to this much larger, highly folded structure. And one of the reasons we're interested is that we think the shape and structure of our brain is really important for its function. And there's a lot of evidence from that in many neurodevelopmental disorders where you get a change in the structure and shape that's forming. We investigate this process using an interdisciplinary approach. We use high-resolution imaging combined with biophysics and some computational modelling to try and understand how these folds are mapped out and what's regulating their development. We've uncovered some of the mechanisms for this, but now we're looking much more deeply into how that pattern is very, very similar between every individual. We've also recently looked at how SARS-CoV-2 can affect fetal brain development and how this may then change structure and then ultimately function later on.
My lab studies the development of the choroid plexus, a brain region responsible for secreting the cerebrospinal fluid and protecting the brain from pathogens and toxins. So to study this region, we use organoids, a human model derived from stem cells. And this region would be impossible to study otherwise because the development happens in utero. So using this model, we can now understand better the role of the choroid plexus in human development and also in certain developmental diseases. We can perform preclinical tests as well to assess the permeability of pathogens and drugs into the brain, which is a major challenge in many clinical trials. And for example, pathogens such as SARS-CoV-2 can infect the choroid plexus, so we need to be able to investigate this and test this to be able to understand also the translational impact in patients affected with COVID or, for example, long COVID.
Alterations in brain development are at the root of many neurological and psychiatric conditions, from intellectual disability to epilepsy to psychosis. But we can only treat what we understand. That's why it's critical that we bring together all our resources to expand our understanding of the developing human brain. At KIDS, we have a unique opportunity to be transformative and to improve the treatments for these conditions. There is no other place in the world with a concentration of experts, facilities, and laboratories to make this a reality. So we can really make transformative impacts with your support.